All right, so I'm going to share my screen with you. Hopefully you can see that. So I did my project on achromatopsia and specifically focused on the CNGA3 and CNGB3 genes. And then on the homepage, I included this YouTube video because it gives a very good overview of the roles and functions of the CNG channels in the retina. It goes into achromatopsia and even possible treatment methods. So I figured it'd be a good overview before you dive into the website. So I just gave a brief overview of what is total color blindness, what are photoreceptors, what are rods and cones, and then what is a CNG channel, just to give a good overview and to avoid any confusion when reading the rest of the website. And then I included this image here to show that you can't really see a difference in the retinas between a normal retina and a achromatopsia retina. So you'd have to use other diagnostic tools. So here are the signs and symptoms. I included this image here so you could see what normal vision looks like and what achromatopsia patients see, which is just black and white. Here is a list of some diagnostic tests that can be run to diagnose achromatopsia. And this is just an image of one of the diagnostic tests, which is optical coherence tomography. And it can be used to visualize the structure of the dysfunctional cones. So this is the structure and the function of the retina. I wanted to dive into this before I went into the development of the retina. So it goes into the layers, where the cones and rods can be found and things like that. And then this image is a good view of what that's talking about. And then here we go into how does a normal retina develop. I did my best to find some things on the role of CNG in that process, but I couldn't find a paper on it. So I wrote down how the retina develops from the pages that I could find. And then I did find a research article talking about where CNG can be found during development in a developing chick embryo, which was pretty interesting because it was present throughout the body during development. And then this is just a simplified view of the CNG channel with the cGMP, which is something that is very important to keep this channel open. And then right here, I just went into how does mutated CNGB3 affect vision, which it causes cone dysfunction and total color blindness. So right here is just a little bit more of a dive into what is the CNGA and CNGB3 genes, what are their role in achromatopsia. And then this image right here just shows a really good overview of the signal transduction system and a normal photoreceptor and then a mutated photoreceptor. And then this is a little bit more into the signaling pathway of CNG. And then this photo is a little bit more detailed on the role of CNG in the signaling pathway to hyperpolarize the photoreceptor. So this just goes into really the only treatment option, which is glasses right now. I included colorblind glasses because it is something that's becoming a little bit more popular, but it cannot be used for achromatopsia because it does not have the power to fix or heal dysfunctional cones. And then possible treatment methods, as of now, there aren't any cures or anything, but this is gene replacement therapy that was done on a CNGA3 knockout mouse, and it was successful. So hopefully this can be translated to humans in not too long. And then for current research, I decided to talk about what happens to retinal function when CNGB3 is deficient. And one of the main findings in this was that CNGB3 knockout mice show cone degeneration. So this figure here shows that. And then how does deficient CNGB3 affect CNGA3? And in this image, you can see that when CNGB3 is knocked out, CNGA3 is severely downregulated. And then I included what's next here because there is a lot known about CNG channels, but the underlying mechanism is still a mystery. We know bits and pieces, but not a whole lot. So there needs to be more research surrounding this channel, as it's not just important for retinal function, it's important for development all over. And then here are my references. Thank you for listening.